Right, good day Junior Techies. This is Mr. P again. We're doing another theory video within the section of cellular respiration. And in this video we're going to be looking at the process of cellular respiration that does take place both inside and outside of the mitochondrial space. So in doing this, hopefully we're going to get a better understanding of how the process of cellular respiration actually takes place, and we better understand to answer the questions that we are going to get at the end of the year. So the first thing to have a look at is cellular respiration as a chemical process, and it is made up out of three different areas or three different processes that all together make up cellular respiration in the aerobic context. So the first one is glycolysis. The second we look at is the Krebs cycle of chemical reactions, and then the third is the oxidative phosphorylation, which all, at the end of the day, add to the effectiveness of the mitochondria, or the body actually, in releasing energy, producing and releasing energy for all of those life processes. So let's just quickly break it down and see what we can remember. So when we look at glycolysis, we start with a glucose molecule. What we find is the first part of cellular respiration is where this glucose molecule is broken down into two pyruvic acid molecules, each of which now are super important to take forward or to progress the cellular respiration as a chemical process. We'll see that in this process of glycolysis, the requirement is a little bit of energy, so we use 2 ATPs and we end up with 4 ATPs at the end of the process. So if we're using 2, we're producing 4, we have a net of 2 ATPs by the end of glycolysis. So you can see that glycolysis is not a very efficient chemical process in releasing energy, but what is super important here is that, one, we've got the pyruvic acid molecules, which become a very important role player in the next phase of cellular respiration, but also the hydrogen ions that have been produced through the glycolysis. They are very important, particularly when we start looking at oxidative phosphorylation. If we look at where these processes take place, and you'll see I've just drawn the outer and the inner membrane very quickly of the mitochondria here. You can see the folds or the cristae of the inner membrane over there, and you can see that inside of this space we have the Krebs cycle and oxidative phosphorylation, and that is where we actually have the Krebs cycle taking place, inside of the matrix of the mitochondria. And you can see there I've drawn the cycle of chemical reactions, or I'm representing them as a cycle of chemical reactions in purple. So my pyruvic acid molecules that have been produced through glycolysis are actually converted chemically to acetyl-CoA. Now I've got 2 pyruvic acid molecules that form through glycolysis, which means I'm going to have 2 acetyl-CoA or coenzyme A molecules that are going to enter my Krebs cycle. For each one of those molecules, the Krebs cycle turns once. So when we're looking at the breaking down of one glucose molecule, we're going to have 2 turns of the Krebs cycle, 1 for each of those acetyl coenzyme A molecules. Once again, those hydrogen ions from glycolysis are only important when we get to oxidative phosphorylation again, and we'll talk a little bit more about them in a minute. Now, with the cycling or the cyclic chemical reactions that incorporate acetyl-CoA, I'm going to be releasing, so through the further breakdown of acetyl coenzyme A, we're going to release, or there's going to be a release of carbon dioxide. Okay, and we know that carbon dioxide is one of the byproducts of cellular respiration, which is quite toxic to human beings. So we'll get to that in a minute, and how the body sort of transports and buffers out those CO2 concentrations. And one of the other products as well of the Krebs cycle is, once again, those hydrogen ions. Now those hydrogen ions are super important role players when it comes to the further metabolism or the further release of energy in the breaking down of that original glucose molecule. So we see that both the hydrogen ions from glycolysis as well as those hydrogen ions from the Krebs cycle are transported to that inner membrane that now has a much larger surface area because of those cristae or those folds. We've got NAD which are carrier molecules which attach those hydrogen ions, and those hydrogen ions then are taken to the inner membrane of the mitochondria where oxidative phosphorylation will take place. Okay, so that is super important because those hydrogen ions are now going to result in the further release of about 36 ATP. That is a much more efficient release of energy where we've got the breakdown of glucose through both glycolysis, which results in the net of 2 ATPs, through the Krebs cycle to oxidative phosphorylation, where I've got another 36 ATPs. So when I've got the full aerobic breakdown of glucose, I end up with a 38 ATP. Okay, so let's look at it in a little bit more of a structured manner. Okay, so here we can see the two membranes of the mitochondria. We know that the outer and the inner membrane are super important in forming the double membrane that makes up the mitochondrial surface area, or the structure. We know that glycolysis is the first chemical process of cellular respiration, and that's where we have the glucose broken down into our 2 pyruvic acid molecules. We're going to release 4 ATPs, we're going to require 2 ATPs, and this is going to leave us with a net of 2 ATPs after glycolysis takes place. And then once again, very important again, those hydrogen ions that are produced through glycolysis. Our carrier molecule, which is NAD, which is going to eventually carry those hydrogen molecules to that inner membrane for oxidative phosphorylation. And because I've got the 2 pyruvic acid molecules, we're forming 2 acetyl coenzyme A's. And this takes place in the cytoplasm. So this is on the outside of the mitochondria, and we're going to have the acetyl CoA and the hydrogen ions move or migrate to the matrix of the mitochondria. So here we're having a look again at the Krebs cycle, one of the important molecules that are being produced or given off as a byproduct of the metabolism of glucose, CO2 or carbon dioxide, and those once again hydrogen ions that are so important when we look at oxidative phosphorylation. NAD being the carrier molecule of these highly energized hydrogen ions, and they get taken to that inner membrane. And this is where my oxidative phosphorylation takes place. It requires oxygen for the purpose of producing water as that final end product. Once the energy carriers in that inner membrane actually release the energy and produce those ATPs as hydrogen is moved from carrier molecule to carrier molecule on that membrane, energy is released, ATPs are produced, and at the end of it, for every one of those hydrogens, they're going to combine with oxygen, and I'm going to have water as my product. Okay, now if we had to quickly summarize this whole section again into the chemical reaction, I'm going to have a net of 38 ATPs after the aerobic breakdown of carbohydrate. I'm going to have the reactants of glucose and oxygen, and that now we know where the oxygen is required in the whole process of cellular respiration. The oxygen is needed in oxidative phosphorylation, and we look at where we've got the products forming. We see that these products, the H2O formed through the process of oxidative phosphorylation as well. The CO2 as the byproduct is produced in the turning of the Krebs cycle, and those 38 ATPs that result at the end of the breakdown of a glucose molecule, 36 of them produced through oxidative phosphorylation and those highly energized hydrogen ions, and then 2 ATP net produced through glycolysis as well. Well, Junior Techies, thanks for joining me for this video. It does feel like we have moved through quite a bit of information quite quickly. I hope that the video has been beneficial for you to watch in better understanding cellular respiration and how all of those processes fit together. Do tune in for some of the other videos in this section as well, where we look at a little bit more of the theory, but also where we practice some of the exam style questions, or those exam type questions, where we do question analysis and we help you understand exactly what it is the question is asking you to answer, to make sure that you can maximize your results at the end of the year. Right, Junior Tickies, if you haven't already, get a hold of that worksheet. As I said before, look at that worksheet memorandum while you work through it, and I do hope to see you again soon. Thanks so much, Junior Tickies.